So we have the good fortune of hindsight is 2020, and we know some of the basis for what Mendel was observing is because of structure of the chromosome that was discovered and worked on for quite a few years in the early 20th century. And we were able to really learn a lot about Mendel's work based on studying of chromosome behavior. And so we're going to jump around a little bit and we're going to kind of jump forward in time to after Mendel has published his work and microscopy got a bit better, staining techniques for looking at cell structures got a bit better, and just a general understanding of the structure got a bit better. And then we're going to come back to Mendel's law of independent assortment and see how that affects the offspring and the inheritance of multiple traits at one time. So our understanding of Mendel's conclusions comes from something called the chromosomal theory of inheritance. And the chromosomal theory of inheritance was proposed by Walter Sutton and Theodore Boveri, and they said that the foundation of genetics is really in chromosomes as the carriers of genetic material. So basically, they gave our alleles and our genes somewhere to hang out. And they said that those genes and thereby those alleles are found on chromosomes. And this was useful because it explained some of the things that Mendel was seeing. Now, this was late 1800s, early 1900s, and keep in mind that at that point, the structure of DNA had not been discovered. Structure of DNA wouldn't come till the 1950s, and so genetic material was a little bit more elusive. What exactly was the mechanism for coding and the structure for coding those genes and those traits wouldn't be figured out for a while. And people knew that there were some proteins in there and eventually some nucleic acids in there, but it would be a little while till they would specifically determine that genetic material is encoded in nucleotides. So for now, we're just going to think about chromosomes in general, not necessarily nucleotides, because Sutton and Boveri didn't know what they were, and of course Mendel didn't know what chromosomes were. But we can use that to help our current understanding of genetics. So the chromosomal theory of inheritance gives us a few key characteristics of chromosomes that allow them to basically be the vehicles that Mendel was talking about during transmission of genetic information. So the chromosomal theory of inheritance describes how our movement of chromosomes during meiosis and fertilization essentially parallels Mendel's patterns of inheritance and the passing on of traits from parent to offspring. Chromosome theory also provides a mechanism that explains Mendel's laws because we can now identify those factors and specifically those paired factors that Mendel was observing in his crosses. So the chromosomal theory of inheritance identifies chromosomes as the vehicle or the location for those discrete traits or discrete alleles that were being passed from parent to offspring. And then finally, the chromosome theory of inheritance says that chromosomes are linear structures on which genes are located, and that those genes have a specific location on those chromosomes. In other words, for a specific chromosome, there's always going to be a specific gene at a particular location on that chromosome. So all together, the chromosomal theory of inheritance says that chromosomes are the basis for all genetic inheritance, and that genes are located on those chromosomes. In other words, chromosomes are the discrete units that were passed from one gamete to fertilization and then on to the offspring. So now that we have an understanding of Mendel's theory, or I'm sorry, Mendel's law of segregation and the chromosomal theory of inheritance, we can put those together and link our separation of alleles that Mendel observed with the separation of homologous chromosomes in meiosis. So this image depicts the alleles on our chromosomes, and it shows the process of DNA replication and then the separation of those alleles. First, separation of homologous chromosomes and then separation of sister chromatids. And so what this tells us is that each one of these 4 gametes has an equal chance of receiving the parental allele. And if we look at the gametes themselves, and it doesn't actually matter which side you look at because it shows the same thing, 50% of our gametes have allele 1 and 50% have allele 2. And the same thing is shown on the other one when there is crossing over, when we take into account crossing over. We have 50% of our gametes have one allele and 50% have the other. So there is a 50% chance of the parent passing either allele. So Mendel tells us that there are discrete units that we see dominance and recessiveness. He tells us that there is segregation of alleles. And then lastly, his last conclusion was that there is independent assortment of chromosomes. So independent assortment tells us that our chromosomes are separated into gametes in any combination. So what this is talking about is when we have 2 different pairs of homologous chromosomes. So in this image here, we have homologous chromosome number 1 that has the R gene on it, and then big R, little r. So those are our homologs there. And we have a second chromosome that has the Y gene on it, big Y, little y. And so independent assortment tells us that we can get any combo of R alleles, the big or the small, and Y alleles, the big and the small. And so we have to keep in mind that all individuals eventually have to have one of each chromosome. So what this is saying is that every individual is going to get one of the R chromosomes and one of the Y chromosomes. And the alleles get shuffled in any combination. And if we look down at the bottom, we can see those allele combinations. So we can get big R, big Y. We can get little r, little y. We can get big R, little y, or little r, big Y. So this shows all combos of 4 alleles. So this leads us to the dihybrid cross. And the dihybrid cross is a demonstration of independent assortment. In a dihybrid cross, we are looking at the results of crossing gametes with 2 different chromosomes and 2 different traits at the same time. So if you do not remember independent assortment from meiosis, you will want to review metaphase 1 of meiosis, where our tetrads line up in any combo or orientation at the metaphase plate. And depending on which way they are oriented, they're going to separate into their separate cells at the end of telophase and then into separate gametes in different combinations at the end of meiosis. And the previous slide was showing that. It just didn't show the tetrads lining up. Keep in mind that we also have crossing over happening, so that's going to increase our variation a little bit more. So we're going to look at these dihybrid crosses. They are going to demonstrate the principle of independent assortment and relate that to meiosis. And the end result, we say that the gametes on these different chromosomes are going to separate independently or sort independently. So I would like you to take a minute, describe the differences between segregation and independent assortment, and even see if you can link these back to meiosis. I have mentioned where they happen. See if you can recall this on your own. All right, so let's get into a dihybrid cross. In a dihybrid cross, we are going to be crossing 2 traits with 2 alleles each. So we have gone back to our round and our wrinkled seeds, and then our yellow and green seeds, which we haven't worked with yet. Round is dominant and yellow is dominant. Which means that wrinkled and green are recessive. So for our demonstration here, we're going to start with the round and yellow true breeding parents. So we've got that true breeding parental generation. And the other true breeding parent is going to be the wrinkled and green. So we're going to cross round and yellow, wrinkled and green. Both are homozygous. When we made our Punnett square for our monohybrid cross, we separated our gametes, and we're going to do the exact same thing here, except that we need to remember that we are doing independent assortment, which means we need all of the combos of R and Y, whichever combo they are. And so you are welcome to do this however you learned it. I teach my students to use FOIL from math. If you don't use FOIL and you have another method, that's perfectly fine. But you want to make your gametes, and they need to be all possible combinations of R's and Y's. Now this one is going to be pretty easy. We get our gametes big Y, big R, that's the only option, and little y, little r. So the F1 generation are all going to be heterozygous for their particular traits that they started off with. And then Mendel did an F1 self-fertilization, so he crossed this with another F1 individual, and he looked to see what would happen. Now, I'm not going to draw out this whole dihybrid right now. I'm going to make the gametes, and then I'm just going to direct you to the picture. So to do the F1 cross, we need to make the gametes. So we are going to take the first two alleles, and that's our gamete 1. We're going to take our outer alleles, and that's gamete 2. Our inner alleles, and our last alleles. So those are our 4 possible gametes, and we are going to put them on a big Punnett square, and then we're going to do the same thing for the other parent. First, outer, inner, and last. And we're going to put those on the Punnett square, and then we are going to perform fertilization. So big R, big Y, big R, big Y, that's going to give us a round and yellow. And I think it would be helpful for you to write this out, figure out your genotypes for each one, and then just check them with this one here. You'll notice that they have given us the phenotype. Remember, the phenotype requires both of those chromosomes. So we need the chromosome that has the color, and we have the chromosome that has the shape. And you have to have one of each. If you don't have one of each, you did something incorrectly with your gametes. So we have one of each, so we get 9 round and yellow. We get 3 wrinkled and yellow, like this one here, 3 round and green, and 3, let's see. Oh, they did this a little bit different. Green and yellow. I put them in a different order. I'm not sure why they did that. But anyway, we get the 4 different genotypes, and we get the 9 to 3 to 3 to 1 ratio. This 9 to 3 to 3 to 1 ratio is standard for the heterozygous cross of the parents. And you'll want to memorize this one as the result of the double heterozygote, because we're going to come back to this in chapter 4. So let's do an example here. If you don't feel you need to do the example, just go ahead and skip through it. The first thing we're going to do is make our gamete. So we've got B is for dark hair, little b is for blonde hair. Dark hair is going to be dominant. It's not quite that simple, but that's what we're going to do today. Same thing with eyes, not quite this simple, but this is what we're going to do just for funsies. We're going to use big G for brown eyes, little g for green eyes, and the first thing we're going to do is make our gametes. So it doesn't really matter which one is mom and dad. For mom, we've got first, outer, outer, inner, and last. And then same thing for the other parent. We've got first, outer, inner, and last. And I'm just going to go through, and I'm only going to narrate a little bit of this. And I like to go through one at a time. When I cross them, I just tally them as I get to them. So this individual is going to be dark hair and brown eyes. So I just tally. This is a new one, dark hair and green eyes. Another dark hair and green. Still tallying the double dominant. We've got a new one here. This one is light hair or blonde and brown. So our other combo. And then lastly, we've got blonde and green. Now you could absolutely write out the phenotype ratio. This is the genotype ratio. This is the one time where I'm not going to ask you to do the genotype ratio, and just go ahead and give me that phenotype. So remember, we want to label them. We have 9 dark and brown eyes to 3 dark and green eyes to 3 blonde and brown to 1 blonde and green. And whatever method you want to use to make your gametes and to tally up is perfectly fine. I tally as I go. Other students will fill out everything and then go one at a time. I would suggest that you just find something that works for you so you keep track of them, whether that is crossing off or checking off your genotypes as you go, whatever works for you. All right, so we can use our giant Punnett square to do our phenotype ratio, but we can also do something called a branch diagram. So a branch diagram is basically going to break down your problem, your dihybrid cross, and give you another option instead of doing the Punnett square. And I like this one because this one uses the product law, and it allows us to look at our dihybrid cross as 2 individual problems. So in our textbook, if you want to look at figure 3.11, it's a little small to put on the slide here and actually be helpful, but I will work this out for you. All right, so what we're going to do is look at the previous problem as 2 separate problems. And so we're going to think about them as what happens if we cross for the R's and what happens if we cross for the Y's. So we're going to break this into 2 smaller problems. Now notice I put an emphasis on and. This means that we're going to use the multiplication rule here. So we are going to look at what happens if we cross for R and if we cross for Y. So to show you what I mean, if we break down our R problem and we just cross our heterozygous big R little r with big R little r, and we look at our phenotypes, we have a 3 out of 4 chance of getting big R with one of our other alleles, and it doesn't actually matter which one for this problem because all we're looking at is the phenotype. And then we've got our other phenotype, 1 out of 4 are going to be little r little r. And then if we do the same thing with our Y problem, we'll recognize that 3/4 of them are going to be yellow and 1/4 of them are going to be green. And so we can look at the chance of any of these combinations. So we can look at the chance of round and yellow, round and wrinkly, green, round and green. We can look at the chance of wrinkled and yellow or wrinkled and green. And so we call this a branch diagram because we're going to start with our 3/4 round, and we're going to say, well, what happens if we want to figure out the probability for yellow? And what happens if we want to figure out the probability for green? So we're going to take 3/4 and multiply 3/4 for yellow, and we are going to end up with 3/4 times 3/4, which equals 9/16 to get a round and yellow plant. Round and yellow seed. And then we're going to say, well, what happens if we do round with our green? Well, that is 3/4 times 1/4, which is equal to 3/16. So 3/16 are going to be round and yellow. And then we can do the same thing for our wrinkly. 1/4 are wrinkly. We can look at wrinkled with yellow, 3/4 yellow, or green. So if we go round with yellow, we have 1/4, keep our order straight, 1/4 times 3/4 is 3/16, which are wrinkled and yellow. Sorry, wrinkled and yellow. And then 1/4 times 1/4 is 1/16 are wrinkled and green. So we end up with the same ratios, the 9:3 to 3:1. We've just taken out the Punnett square, which for some of you is going to be much, much easier. For some of you, this is going to be far more intuitive. For others, that Punnett square is going to kind of be a crutch. You can just always go back to it. I will tell you that on an exam, I will ask you to do a branch diagram instead of a dihybrid cross. It will probably ask you to do both of them, or at least interpret a dihybrid or set it up, but you will have to show me that you understand the principle behind the branch diagram. So just kind of a quick summary, a branch diagram is going to allow us to figure out the phenotype probabilities or ratios without a Punnett square. So in the previous problem, we got 9 out of 16, 3 out of 16, 3 out of 16 again, and 1 out of 16. And that represents our 9 to 3 to 3 to 1. And then, of course, if you're writing out the ratio, you're going to want to label it as well. So it might help you to make a small Punnett square for each of the two traits. If you don't need to do that for each of the individual problems, that's perfectly fine. You can do it in your head if it makes sense to you. But I do want you to show me your branch diagram, at least the math. Even if you're not drawing the branches, please show me your math. Please don't do all of the math in your head. I want to see what you're doing. So we're going to use the product law because these are, excuse me, two independent events. The inheritance of the R chromosome is independent from the inheritance of that Y chromosome. And that's why we can use the multiplication rule, because we are looking at the inheritance of R and Y chromosomes. So this slide here just shows you a nice, neat version of what I just did. There's nothing new here. It's still the round, wrinkled, yellow, and green, but it looks much neater and you can see it a little more clearly. So we have two examples here. We already did the Punnett square for the blonde and green-eyed cross here. And so you already know that this should give you the 9 to 3 to 3 to 1. And you should actually recognize this because this is our full dihybrid for both traits. So you should recognize this is already going to give you a 9 to 3 to 3 to 1. I put this one in here so that you can do the branch diagram on your own and just verify that you get the 9 to 3 to 3 to 1 and that you understand what is happening there. So go ahead and pause and do this one for practice. And then we have a second example that I would like you to try. I would like you to fully work this out, figure out all of the phenotypes and genotypes, and at the end, you should end up with a 1 to 1 to 1 to 1. If you feel like you need a Punnett square practice, you can also use this one for Punnett square, but I would like you to definitely try it for the branch diagram as well. If you were watching this in class, you would do this in class with your table, the people at your table, and practice this. So if you would like to do this and then check it with me, that's fabulous. If you get the 1 to 1 to 1 to 1, you probably don't need to check in. But if you don't and you have questions, please come into office hours. All right, so being able to use a branch diagram and recognizing that any of our combinations of chromosomes are just the product of the individual probabilities is going to help us if we get to something far more complicated. So I'm guessing that none of you want to do a tri-hybrid cross. This becomes completely ridiculous very, very quickly. And so instead of doing that, we can use probabilities to figure out the chances of any, or the ratios for any particular cross. Now on this screen, I'm not going to do this triple branch diagram, but basically what you would do is draw out AA cross AA. You have 3 out of 4 dominant, 1 out of 4 recessive. And then from there, you're going to branch to your B options. And from there, for each one of these, you're going to branch to your C options. Okay, so this is going to get really big. And then you're going to do it for little a, little a. I would suggest that you try this. I will leave it up to you. If we have time in class, I always do this one. If we're kind of running out of time, I may not do it. But the other thing that I would like you to recognize here is what you could say is we're going to cross these parents. What is the chance of getting a dominant A, recessive B, and dominant C phenotype? Well, then you can look at this and say, well, based on the individual Punnett squares, we have 3 out of 4 times 1/4 times 3 out of 4. And then you can very quickly figure out, well, what is the chances of being dominant A, recessive B, and dominant C? And we would determine that this is 9 out of 64. And then you don't have to do a 64 square Punnett square. I don't think anybody wants to do that. All right, so I got a little bit ahead of myself. This is just breaking down the problem for you. There's nothing new here. This is just a summary.